This last company presentation will be given by Anders Kronström, who is the CEO of Biosergen, who does antifungals.
Thank you very much. Yes, I'm Anders Kronström, the CEO of Biosergen. And thanks a lot for the invitation and the opportunity to talk about Biosergen and some of the highlights we have in our development program. As the title suggests, we are fighting severe fungal infections, and we focus on infections that requires systemic treatment. These are typically very serious conditions, and the severity of these conditions are well illustrated by typical mortality numbers that you see on the two most common causes of fungal infections that you see in these slides. So the two most common ones are infections by Candida and infections by aspergillosis. Normally, patients that get these kind of infections are patients that have a suppressed or some kind of a compromised immune system. The current drugs that are used have several limitations in terms of efficacy and toxicity, but also an increasing problem with drug resistance. That means that the infections by strains that are resistant to drugs that are currently available for a clinician. So we definitely think there's an opportunity. There's a need for new drugs and better drugs, improved drugs for the market. A few words on Biosergen. Biosergen is actually a Norwegian company. It was founded in 2004 by Institutet Venture and Karolinska Institute Holding. Sintef is a foundation, is the largest research organization in Norway. And the company was founded based on research generated by the Norwegian University of Science and Technology located in Trondheim in Norway. So what is this innovation all about? There was some clever group of scientists at the university that worked together with Sintef, and they found a way to modify the gene cluster that is responsible for the biosynthesis of nystatin. Nystatin is a polyen. It's a known antifungal agent, has been known since the 50s. And by doing these modifications, they were able to generate new leads and new hits that later on was screened for antifungal activity. One of those agents, BSG005, showed very promising data and was selected as the lead CD and is now in preclinical development. So what data do we have on this compound today? Well, in terms of in vitro, we have performed a rather broad spectrum screen for antifungal activity. It's around 250 different strains, and we can show a very broad spectrum of efficacy. Possibly even more important is that we see that we have a very strong fungicidal activity of our drug, and that is superior to other comparators whose activity is largely fungostatic. And this could, in the clinic be very relevant and a very important feature, obviously. In terms of in vivo data, we have tested our compound in immunocompromised mice models. It's a very common model for antifungal agents. We can see, and the efficacy is measured in terms of overall survival of infected mice that are treated, and also looking at fungal burden in specific organs. And we can see some very promising data. We have a much better effect than amphotericin B in the form of ambisome, which is a formulation of amphotericin B. And also we have very good efficacy compared to compounds from the most widely used class of drugs that are used today, the azoles. So we have tested for fluconazole and voriconazole. Generally speaking, toxicity is quite an issue in this disease area. And for that reason, we have done specific tests for toxicity. In acute models, when we have compared ourselves with amphotericin B, we can see a significant reduction in toxicity, 5 to 10 times in single-dose studies and in studies when dosing has been repeated for up to 10 days. And we have also looked specifically for urine enzyme markers, and again we can see less toxicity. The polyen in particular are sometimes or commonly associated with nephrotoxicity. That is one of the limiting factors for using these drugs. So with this data, how would we then position our compound? This grid shows strength and weaknesses for BSG-005 and some of the key competitors from various drug classes. To summarize, we see two key opportunities for BSG-005. One is that we think we may have a better therapeutic window or a dose window compared to existing drugs. That means that we can go up higher in dose without being limited by the toxicity seen with some of the comparator compounds. The other opportunities is in the drug resistance area, that it's possible that we could be effective in infections caused by strains that are resistant to drugs currently on the market. A few words on the timelines. We are in preclinical development. 2016, this year, we focus a lot on drug development activities that will take us up to clinical studies in 2017. We do a lot of chemistry work and formulation work, and we're also doing a lot of toxicity studies this year. After the summer, we need to ramp up some of the regulatory work and also work more with clinical expertise to design our studies. So we hope that in 2017, we will start dosing our drug into man for the first time. And if all goes well, we could start proof of concept studies in patients in 2018. I also like to mention that we are active in China. We established a joint venture last year in China, and our overall program will support activities in China as well. And the aim is also for the China program to start dosing sometime next year in 2017. With that, I like to conclude. We think we have a very promising compound. We are rapidly moving it forwards in terms of development, and we have a very good plan in place to take us up to first-time-in-man studies during next year. I'd like to take the opportunity to ask a question to this audience. If you've heard anything or seen anything that made you think, can you get involved? I mentioned that we have a need for regulatory and clinical expertise this year. And going forward, supporting the program beyond first time in man, we need funding. We need people that are willing to share risks and costs, and obviously also reward in the end in terms of supporting the program long term. With that, I thank you for your attention and time.